For decades, Alzheimer's disease has been one of modern medicine's most profound and, well, heartbreaking puzzles. We've known the main culprits causing damage in the brain for a long time, but a huge piece of the puzzle was always missing. Today, we're diving into the incredible research that finally found that missing link.
So for years, the story of Alzheimer's really revolved around two main villains. Scientists knew they were connected, but they had no idea how. Let's think of this like a detective story, and we've just been handed the case file on our two prime suspects.
All right, so on one hand, you've got these things called amyloid beta plaques. Picture these as sticky, nasty clumps of protein that build up in the space outside of our brain cells, our neurons. And then, on the other hand, you have tau tangles. These are these twisted knots of a different protein that form inside the neurons. These two have always been the classic, tragic hallmarks of the disease.
And that, right there, brings us to the central mystery that has stumped researchers for ages. How on earth does a problem brewing outside the cell, the amyloid plaques, manage to trigger a complete disaster inside the cell, the tau tangles? You know, how does the bad guy on the street corner get someone inside the house to start wrecking the place? That's the question we're finally going to answer.
The first huge breakthrough really came when scientists decided to look somewhere new. They stopped staring into the empty space between the cells and started zeroing in on the cell's actual border, its surface. They started hunting for an accomplice, right? A weak point, a receptor that might be letting the toxic message from amyloid beta sneak past the neuron's defense. And you know what? They found a prime suspect. It's a receptor protein with a bit of a mouthful of a name, Fc gamma receptor IB. We're just going to call it FCGRIB. Now, the best way to think of this thing is like a docking station on the neuron's surface. It has its normal day jobs for sure, but researchers started to suspect it had a dark side, that it was the very port where these toxic amyloid proteins were getting a foothold. And this led them to a really powerful hypothesis. Maybe amyloid beta wasn't actually breaking and entering. What if instead it was just docking at this FCGRIB receptor on the surface and using it, like a mole on the inside, to send a toxic signal into the cell, a signal that would kick off all the internal chaos? Okay, so with a suspect in their sights, the next step for researchers was to map out the entire crime. What they uncovered was this incredibly precise, step-by-step toxic chain reaction, a series of molecular dominoes just falling one after another, leading to disaster. Let's walk through it. So let's trace these dominoes. First, the toxic amyloid beta from outside locks onto that FCGRIB receptor. That acts like a light switch. It activates the receptor. Second, once it's activated, the receptor calls for backup. It recruits its inside accomplice, an enzyme called SHP2. Third, SHP2 gets to work and messes up the cell's internal chemistry, causing a big buildup of a specific signaling molecule. And that buildup, that's the final, fatal domino. It is the toxic signal that tells the tau proteins to go haywire, form those destructive tangles, and ultimately kill the neuron from the inside out. That is the entire chain of command, from the outside threat to the inside catastrophe. Now, a great theory is, well, it's just a theory until you can prove it. To really crack this case, scientists needed undeniable hard evidence, and their strategy for getting it was brilliant. If this chain reaction really is the killer, what happens when you break the chain? The strategy was really simple, but so elegant. If this chain of events is truly what's causing all the damage, then what happens if you just remove one of the links? So they tested this idea using special mouse models, mice that were specifically bred to develop all the symptoms of Alzheimer's. And the results were, frankly, stunning. As you can see, the mice with Alzheimer's symptoms, they had pretty significant memory problems compared to the healthy mice. No surprise there, but, and this is the amazing part, when the scientists got rid of that FCGRIB receptor, breaking the very first link in the chain, the memory problems just vanished. The mice performed just as well as the healthy ones. Incredible. So then they tried breaking a different link. They thought, okay, what if we block that inside accomplice, the SHP2 enzyme? And again, the results were just incredible. This chart shows that when they blocked SHP2, the amount of those toxic tau tangles inside the neurons of the Alzheimer's mice, it plummeted. It was almost down to the levels of a healthy mouse.
The proof was getting really, really strong. Okay, so the mouse data was powerful. I mean, it was a huge deal. But the most important question of all was still hanging out there. Does this exact same thing happen in people? Is this FCGRIB SHP2 pathway the real culprit behind human Alzheimer's disease? And by examining brain tissue from deceased patients, they got their definitive answer. And the data, it's just crystal clear. In the brains from healthy people, or even those with just mild cognitive issues, that receptor was dormant, it was quiet. But in the brains of patients with full-blown Alzheimer's disease, the receptor was switched on, it was activated. And that activation lined up perfectly with the presence of tau tangles. The smoking gun they found in the mice, it was now confirmed in humans. Case closed. So look, this discovery is so much more than just solving a cool scientific puzzle. By identifying this precise molecular pathway, this discovery gives us a highly detailed roadmap for developing brand new treatments. It literally opens up entirely new avenues for fighting back against this disease.
And this roadmap reveals multiple, very specific points where we could intervene. So instead of just one single target, we suddenly have options. We could develop therapies to block the initial handshake between amyloid beta and the receptor on the outside. Or we could focus on stopping the receptor itself from sending its toxic signal. We could even develop drugs to inhibit that SHP2 enzyme, stopping the whole cascade from inside the cell. It's a whole new playbook.
The researchers themselves summed it up perfectly in their paper. They said, the FCGRI-BB-SHP2 signaling axis provides the missing link between A-beta and tau pathologies. It's not just a link, it's the missing link. The bridge that connects the problem outside the cell to the disaster inside. And this incredible discovery leaves us with this powerful and deeply hopeful question. For decades, the fight against Alzheimer's has felt like an uphill battle. But now that we have the entire toxic chain of command laid bare, could finding a way to break just one of these links, could that be the key we've all been searching for to finally stop the damage and preserve the memories of millions?